Good afternoon, everyone, and welcome to the 2023 Ian P. Howard Distinguished Lecture Series. We've been holding this lecture series for over 15 years now. It's in honor of Ian Howard, who was the founding director of the Center for Vision Research here at York University. In the 1960s, he founded the York Vision Group, which then grew into the Center for Vision Research, and we now have over 40 faculty and 5 faculties. And I believe 8 or 9 departments. So it's grown considerably. Lots of HQP and trainees have gone through. And this particular series is in honor of Ian, the founder, who's a very interesting and eclectic man. He was my supervisor for my PhD. But he's very well known for writing 3 books, one on spatial orientation. Then 20 years later, he had to narrow it a little bit because the field grew so much on visual orientation. And then 3 editions of his book on monocular vision and stereopsis. Very active experimenter, creator of unique apparatus and other things. He's fondly remembered by us here at York. Sadly, his wife Toni died this year as well, so we've lost both the Howards in the last 10 years. That said, it's my great honor to welcome Jack Gallant. He's a Chancellor Professor at UC Berkeley. He has appointments in many departments, so he's a good example of the sort of thing we try to do here at CVR. He's got appointments in psychology, neurology, electrical engineering, and computer science. So very interdisciplinary. He's a senior member of the IEEE, and he's currently chairing the IEEE Brain Community.
That was last year.
That was last year. Oh, apologies.
So if you have an IEEE brain thing, don't email me.
His research is really interesting. We saw some of his work on V4 yesterday, but really he's looking at high-resolution functional mapping of the brain, really looking at trying to understand function, go beyond description and understanding. He's created the most detailed current functional maps of human brain networks for vision, language, and navigation. And he's going to talk to us today about navigation. He's also, I found out last night, a very charming and entertaining dinner companion, and I think he's going to give us a great talk. So, welcome, Jack.
Thanks very much. Thanks for that kind introduction. The last time I was at York was 2005, and both 2005 and before I showed up this week, I always assumed that York had been around for like 200 years, because the vision program at York has always been at the front of all the best vision programs. You guys have been really, really consistent, and everything out of York is fantastic. And it's the variety of work going on here. I'm so happy to see that as the generations have turned over, that you guys have been hiring new people and keeping the CVR running. It's really fantastic. I just cannot believe this place was only built in 1960. It's just astounding to me. Really fantastic. So I'm very honored, given that you are a great group of vision people, to be giving this talk, because I'm really a vision guy. All my training in graduate school and my postdoc was all in vision, and I did nothing but vision up until about 2008 or so. And then I started branching out to other kinds of problems. But today I'm going to be talking about what I do consider a vision problem, but it's a vision problem that touches on a bunch of other cognitive systems, which is navigation. So humans, we don't navigate by olfaction, at least as far as I know. We mostly navigate by vision. And so vision, the visual system, is intimately tied into all of the navigational systems of the brain. And navigation provides a good platform for looking at vision and cognition and their linkage together, and also for looking at visual motor interactions. And we're going to explore all of that in this talk. So I should just mention briefly that the reason that this navigation project is a big project spearheaded by one superhuman postdoc who was a graduate student with me and is now a postdoc, Jianzhao Zhang, who's been working on this project for about 6 or 7 years, and not even one paper has been published on this yet. Not Zhang's fault. You'll know why when I show you the data. There's a lot of threads that all have to come together, and we're now writing all these up in a series of papers we're going to publish. But this work, Zhang's work, actually grew out of an earlier project that started back in about 2010, where we started trying to, we wanted to set up the infrastructure, both in terms of the experimental infrastructure, in terms of the modeling infrastructure. We would need to do virtual reality in the MRI environment. And that turned out to be a much harder and more complicated and larger problem than we thought. So we really did no virtual reality experiments after our pilot experiments from 2010 till about 2018. When Zhang joined the lab, we felt like we were close enough to begin to start collecting data, even though we would still have more development work to do. And we had to decide what kind of problem to work on in virtual reality. And we all concluded that navigation was probably the best target because it's a principled problem where there's a lot of animal data that we could leverage to try to make sense of what was going on in the human brain. So that's where this project comes from. And if you guys want to hire somebody who's going to be the next, superhuman MRI guy, this is the guy to get. Get him now before papers come out, when he'll be in high demand. All right, so I promise the papers will come out eventually in Jack time. They'll be there. All right, so there's a lot of prior work on navigation that has been done in rodents and a lot of work in humans, some work in primates, not so much, most of it in rodents and humans. And this kind of summarizes in a very cartoony way what is known about the navigation systems in the human. You can see there's a small sort of sensory, a small network on the right of navigation-related areas, the hippocampus and entorhinal cortex, the prefrontal cortex. The OPA, PPA, and RSC, these are three of the core navigation areas in traditional MRI navigation experiments. OPA is the occipital place area, PPA is the parahippocampal place area, and RSC is the retrosplenial cortex. And these networks interact with sensory, these areas interact with sensory inputs and memories from the hippocampus and plans from the prefrontal cortex to produce the motor actions necessary for navigation. I took that slide out. So I do want to mention that the work that sort of recovered this network in humans mostly uses reduced paradigms. So for example, you'll do an MRI experiment where you'll show somebody a place and ask them if it's familiar or not. Or you'll show a picture of a place, and then you'll show the picture of a place taken from a slightly different camera angle, and you'll ask if it's from the same camera angle. Or you'll show them a sequence of static images that represent the turns they would make in a path that they have learned previously. These are all very highly reduced, very highly simplified navigation environments. And one of the big concerns in my lab, or one of the big focuses in my lab has been to work on naturalistic stimuli and naturalistic tasks. We want to get as close to the real world as possible because since the brain is a nonlinear dynamical system, the only way we can be sure that the model we build is going to generalize to the real world is if we build that model in the real world. So that's kind of just an underlying philosophy of my lab. So based on that, we decided to kind of jump over all the traditional way people had done things in the navigation world and do an experiment in virtual reality. So the first thing Zhang did is he built a giant town in Unreal Engine. Unreal Engine is a graphics engine that allows you to build virtual reality environments. And because these engines, these virtual reality environments are now used to train autonomous vehicles, there's actually an abstraction layer above Unreal Engine called Carla that allows you to implement autonomous vehicle algorithms in these virtual environments. And then we built another abstraction layer on top of Carla that allows you to drive in the virtual environment with other autonomous agents that are driving in that environment. Okay. So we built a town that's about, I think, 3 by 5 kilometers on a side. There are about 200 landmarks, roads, and buildings in this town that people have to learn. There's countryside, suburban, urban, city areas. You can see these look decent. This is kind of PlayStation 5 quality, right? It takes people about 10 hours to learn this map. They learn this outside the MRI machine over a period of weeks. We tell them to just drive around randomly until they learn all the landmarks, okay? Then we put them in the MRI machine. You can see here that we built a few control surfaces here. We have a magnet-safe steering wheel that we built and magnet-safe pedals that we built. This is a fun project. The magnet doesn't like video game controllers inside of it, but you can always outsmart the technology if you want to. And now you can see that Zhang is lying in the MRI machine. He's got the little steering wheel, he's got the pedals, and he's got this optical apparatus that's going to allow the virtual reality seamless to be played into his eyeballs while we scan him, and we're going to put him in the board of this MRI machine. What's he going to do in the MRI machine? He's going to do an Uber driver task. We put him somewhere randomly in the map. We tell him, drive to the grocery store. And then he drives to the grocery store. And then when he gets to the grocery store, the abstraction layer knows, oh, you're near enough the grocery store. Okay, you're done, you've arrived. Now, drive to the gas station. And you just do that. You do that all day long, just like any other Uber driver, taxi driver, for as long as you can stand it. We typically try to collect as much data as we can. In most of these studies, we had maybe 3 or 4 2-hour sessions over a period of weeks. We do some behavioral testing to make sure that people know the maps. Some people are very bad at navigation. We do have some graduate students in my lab who are brilliant people but never could learn the map. We also have one Italian postdoc who is an agent of chaos in this simulation, refuses to obey the traffic laws, and his brain activity is just as mixed up as you would think from a person who drives that way. So there are a few outliers, but generally this task worked for most people. All right, so I always like to begin, hopefully this is a movie. Is this going to show a movie? Uh-oh. Let me show the movie. The movie's the best part. Let me see if I need to click on it. Oh, okay. I'm going to show this movie. I will show this movie later. I want to show it now. I'll show it later. I'll show it at the end. Okay. So basically, we've got a brain. The brain is inconveniently folded up inside the skull. So what we're going to do is we're going to flatten it out and make kind of a large brain-sized pancake. And we're going to project the brain activity on this flat map while people are driving around this environment. And this movie was supposed to show you people driving the environment and the brain activity. It's an awesome movie, and I hope I can show it to you later. But for now, I'll just skip to the static version, which I always put in as a backup in case my movie fails. And this is just 4 frames from this driving experiment. You can see here the person's going straight to orient you on these flat maps. The middle of the map is the visual system. At the back of the head, the far left and far right are the prefrontal cortex. The motor and somatosensory strip is kind of at the top in the middle on both sides, and the auditory cortex is kind of at the bottom. Again, the temporal lobe is down here. So you can see when the person's going straight and traveling behind a car behind them, there's a small amount of brain activity in the visual cortex, but not a lot of other stuff going on. When the person is nearing a stop here, it looks like maybe there's some activity in visual cortex, retrosplenial cortex, a bunch of stuff in prefrontal cortex, maybe some stuff in the somatosensory motor strip. Here they're making a turn. When you're making a turn, of course, you're using the control surfaces, so you get a lot of brain activity in the motor cortex associated with hands and feet. And this is just another example. So you can see that as people are doing this task, there are very rich, complicated patterns of brain activity that are distributed basically everywhere over large parts of the brain. And I like to show these data because these are essentially pre-processed MRI data. They're raw in the sense that the noise has been removed, but we haven't done anything else to model the data. And this gives you a sense of what the problem is. Okay, people are navigating. They're driving in this virtual environment. They're perceiving the virtual environment. They're making decisions about upcoming traffic and pedestrians that might dart out in front of them. They're deciding on what the best route is. They're keeping track of how far they are on that route. And they're using the control surfaces. And all of this stuff is going on in the brain simultaneously. And since we're all neuroscientists here, we assume the soul is irrelevant. That means that all parts of that task must be represented by something on these flat maps, right? So now you can see this is a giant multiple regression problem. I have all of these visual, cognitive, and motor features that are kind of my X variables, and I have all this brain activity that are my Y variables, and I want to regress the X's on the Y's and make inferences about how the brain operates in this task, right? What information is being represented where? Okay. So this is these are just bold activity maps to show you that this task actually does elicit brain activity. This is 3 different subjects. You can see that it elicits a lot of brain activity around the visual cortex, the IPS, intraparietal sulcus, which is a dorsal stream complex of areas involved with attention and eye movements. A lot of stuff up here in these hand representations of somatosensory and foot areas, frontal eye fields, and some other, some more dim spots, but they will be important later, that appear to be scattered around, say, prefrontal cortex and auditory cortex and so on. Oh, there's also sound in this experiment. I didn't mention that. But it's not of interest today. All right. So now we got this task. The cool thing about this video game environment is we built it. We have all the metadata. We know where all the surfaces are. We know where all the other autonomous agents are. We know where all the buildings are. We know what the semantic tag is for everything on the screen. We're tracking the person's eyes while they're in the MRI machine. We're tracking their motor behavior. So we have all of the motor signals and all of the visual signals, and we can put all that together and leverage it. So this is just to kind of make it clear what's going on here. We've got our video game. We can extract, say, I can't remember if this is luminance or distance. This is maybe surface normals. This is probably the semantic category, like cars versus roads versus sidewalks. And we have all of this data simultaneously. Okay, so we're going to use all of this data in our regression framework. I'm going to do this. So in my lab, in our MRI experiments, the most common method we use is called voxel-wise encoding models. So we're going to build an encoding model that relates the X variables, which in this case are the cognitive variables, the motor task, and the visual variables, to the brain activity, which is our Y's. And that's going to be a giant multiple regression equation. And the way we do this is we have some training data, and we have some training features. Now, what are these features? These features are essentially linear or nonlinear projections from the original X variables we started with into a feature space which we hypothesize is important for this task. So for example, we might track the eyes. Eye tracking is probably important, so that's going to be one of our feature spaces is just going to have eye tracking. Retinotopic motion energy, which is motion energy adjusted for where the eye is. That's probably going to be important, so we'll include that as a feature space. Frame-wise motion energy, you can see here's a bunch of navigation variables, navigational affordances, which is can I drive on it, or am I not allowed to drive on it, although that does not apply to Italians. Spatial semantics, which is the semantic structure, the semantic tag for the category for all the objects. Scene semantics, which is just what is it, a countryside scene or urban scene. Attended semantics is the semantic category of the object that you were looking at each point in time, which turns out to be very important. Depth, and so on. You can see all of these. We have 34 variables here that cover all possible kinds of feature spaces that you could extract from this task. Okay? And we're going to fit all of them simultaneously. So we take all of our data, take our brain data, which consists of about 100,000 voxels, each about 2 by 2 by 2 millimeters, by a time series of maybe a few hours. We have our training features, which are going to be 34 different feature spaces. And what we're going to do is we're going to use an algorithm that we developed called banded ridge regression, which is just a variant of Tikhonov regression. It's very efficient and it runs on GPUs, so it's very fast and allows us to solve these problems. And we're going to basically find for each one of the feature spaces and for each one of the features in the feature spaces a weight that relates the brain activity in each voxel to that feature. So like when there's a dog in this part of the display, you know, what is the weight of this voxel for that dog, right? Conceptually, this is really simple. You don't have to worry too much about how it works. I should mention there's 34 feature spaces and about 25,000 features. So we're doing a regression of 100,000 voxels against 25,000 features. And in the old days, that would have been impossible, but you know, statistics and math has advanced a lot, so we can solve that problem. And then after we get those feature weights, we're going to take a new set of data, and we're going to multiply whatever brain data we get by the weights, and that's going to give us some predicted brain activity. And sorry, we're going to multiply the stimulus and task features by the weights. That's going to give us some predicted brain activity, and we're going to compare that to our observed brain activity in a completely held out separate test case. Now, these data do not have repeats. If you've been at some of my other talks, you've heard me talk about the noise ceiling, which is, you know, you build a model, you always want to know how good is this model relative to the best possible model I could ever have, given the data that I have. To calculate the noise ceiling, you need repeats. We don't have repeats in this task because they're driving around each street bespoke. So we can't calculate a noise ceiling. So we're just going to get predictions out, and we can look and see if they're statistically significant, but that's all we can say. All right. Oh, this is just, I don't think anybody here really cares about banded ridge regression. This is, but I just want to mention, this is just to remind me, these different feature spaces, like say the semantic content of the features versus gaze position, they may have different signal to noise characteristics. The brain may represent some of these feature spaces really well, and so it needs very little regularization. The brain may represent some of these other feature spaces poorly. They need a lot of regularization. But there may be more samples in one feature space than another. So every feature space has to have its own alpha, its own regularizer. So this banded ridge regression is just a framework that allows us to estimate a separate alpha for every separate feature space. simultaneously along with everything else. All right, now if all of this seems insane, I just want to point out, this is just a normal experiment. It's just we've shuffled some of the deck chairs on the Titanic, right? So we've got, normally when we do an experiment, classic psychology, we select a stimulus or a task in some subspace of interest, we conduct our MRI experiment, we use the GLM to build a group model, and then we interpret the results. That's a normal MRI thing to do. Here, first we conduct a naturalistic MRI experiment, then we project the stimulus task into feature subspaces, which is analogous to this, right? And then we use the GLM to obtain individual models, and then we interpret the results of the experiment. So we're going to build a model for each voxel in each individual person completely separately. And we have 6 subjects, so we're going to replicate this experiment 6 times. Oftentimes people used to ask me, why in primate neurophysiology can you just have 2 primates, whereas in psychology we need 60 subjects? And the answer is, if you do 2 primates, you're doing 2 separate experiments. So you did the experiment once, and then you replicate the experiment. If you're in psychology and you do 60 subjects, usually you group all those data together, you only do the experiment once. So it actually ends up being better to only have 2 subjects if you analyze them individually than it does to build a group model of 60, because you don't have any out-of-set tests. And the one thing we've learned from data science over the past 20 years is always separate your fit and test data sets. It's the most important thing. Sorry, little mini rant. Okay, but now back to reality. Now, at the end of this procedure, we're going to want to recover, not just like, we don't want to just know where in the brain is, this semantic feature represented, where in the brain is the navigation signal. We want to know sort of what are the navigational networks that we can pull out of the brain. Like, are there networks for low-level vision, high-level vision, navigation, what? And network analysis in MRI usually goes through this method called functional connectivity, where you look at the correlation between the bold signals, the brain activity signals, and you use that correlation matrix to try to infer network connectivity. That turns out to be a fraught method because correlation is not a model, it's just a metric. It has no way to distinguish signal from noise. It's just a metric that works if you have 2 independent variables and your noise is Gaussian IID noise. In MRI data do not satisfy those assumptions. MRI data have a lot of correlated noise that has to do with the susceptibility artifacts that are caused by the fact that MRI signals are higher in some regions of the brain than they are in other regions of the brain, usually because of the presence of a nearby ventricle or an air sac or bone or something like that. So there's a bunch of correlated noise in MRI, and that correlated noise profoundly and inevitably contaminates the results of all functional connectivity analyses. So we came up with a new method that works really, really well, which we call model connectivity. And I like it because it builds on the encoding model framework, but it's dumb as a bag of rocks. So there's 2 good features there. First, you build your encoding models. So remember, in this particular case, for each subject, there's 100,000 voxels, there's 100,000 encoding models. Each encoding model is going to be a vector of 25,000 weights, where those weights are divided across 34 feature spaces. Okay? So now, if every voxel is just a vector, I can just correlate these vectors and create a dendrogram through cluster analysis that basically tells me about the similarity of the vectors. So basically, I'm just going to cluster the voxels according to the similarity in the way they encode the features in this experiment, ignoring their locations in the brain. And then once I have those clusters, I can project them onto the brain to try to find out if they make a coherent story. This method works really, really well. And there's a bioRxiv paper that you can download if you want to hear all of my rants about this. Oh, that's weird. How did that get in there? Extra slide. Oh, this is just to show you the difference between functional connectivity versus model connectivity in a story listening task. So this is not a navigation task. This is just a proof of concept. People listen to stories. We build a semantic model, 1,000-dimensional conceptual model. And if we do functional connectivity, we end up with areas basically everywhere that really don't make much sense. But if you do model connectivity, you end up with really nicely delineated areas that only occupy the parts of the brain that are actually relevant to this conceptual narrative comprehension task. Okay. So now we're going to do model connectivity. I told you what the data was like. We're going to do model connectivity on it, which means we're going to get a dendrogram, right? In a dendrogram, we have to decide where to cut off the dendrogram. And it's kind of an arbitrary decision. That, you can see there in this particular graph, we go from one cluster up to 2,000 clusters. And what you can see is after about, I don't know, 10 or 12 clusters or so, this graph kind of flattens out. So because we want to actually have an interpretable result that we can understand, doing a cluster analysis with 200 clusters wouldn't make much sense. So we're just going to cut it off at like 10 or 12 or something. We'll see in a second. These are pretty low. All right. So these are the data. Now, this is a complicated graph. You're going to see a lot of these, so I'm just going to go through this slowly. On the x-axis here is the 34 feature spaces we talked about earlier. Right? These are divided into feature spaces related to fixation, feature spaces related to low-level vision, high-level vision, motor responses, motor plans, rather than actual physical motor controls, tracking your progress in the map, you know, how far am I toward my goal, goal-related representations like the destination grid representation or the destination anchored vector. So, you know, you can have basically, when you're navigating, one of the things, if you're a good navigator, that you keep track of is where is my destination relative to where I am at all times, what direction, and probably where it is relative to your heading and so on. All those things are represented somewhere in here. So you can see these are divided up into a whole bunch of features, feature spaces. We have in this solution 10 clusters, and now these size of these circles tells you essentially the importance of that feature space to this network, right? So this network 1, you can see that the scene structure and the attend visual semantics are important feature spaces for this cluster, but that the eye tracking feature space is not important, right? Pretty clear. Okay, so now we can use this to try to interpret the clusters that we're getting out in terms of the feature spaces that are represented in each of these networks. And I've kind of done that work for you here by projecting the clusters back onto the surface of the cortex. So there's a low-level vision cluster, which you can see activates the retinatopic visual areas. You notice that, for those of you who remember your MRI retinotopy, you notice that these parts where we're highlighting the most activity, these are regions beyond, outside the retinatopic map. And you may wonder, are those visual areas? Yes, because basically when people are in this navigation task, they're moving their eyes. And so they can move to the edge of the screen, the far left or the far right. So the stimulus is effectively larger than the retinatopic mapping stimulus that we use to map retinopathy. So there's low-level vision, there's high-level vision, which activates all this mosaic of semantically selective high-level visual areas like the fusiform face area, the occipital place area, the exostriate body area, all of those constellation of many, many dozens of functionally specialized semantically selective areas. Then there's a visual attention network. You can see that this is weights most highly on the IPS, the intraparietal sulcus, a bit near the frontal eye field, so it's probably an attention network. And also, of course, I'm not only interpreting this visual attention network in terms of where it projects on the brain, but I'm interpreting in terms of the feature spaces that are loading on each of these maps, right? I have more data that I'm showing you here, in other words, which is the data that I showed you in the last slide. This is a foot area. This is and this is the right foot. This is the left foot. So left foot is going to be the brake. There's going to be a right foot that's going to be the accelerator. There's a hand network for steering, and there's a supplementary motor network that is activated under a lot of manipulation of controls. Okay, these are all fine. This is vision. This is motor. Okay, great. We've done the vision part, we've done the motor stuff. None of this stuff is spooky or scary. It's all like what we should expect to see. But you know, the goal here is to plot navigation networks. So where are they? Well, what in the heck happened there? Oh no, I know what happened. Okay, wait. I have to do some slide for just 2 seconds. I'm just gonna delete these. Okay, so there are some navigation networks. The navigation networks are going to be, I think now, well, what actually, how many next one? 1, 2, and 9. There we go. Okay, good. So if we look at cluster 1 loads very, very highly on all of these navigation-related feature spaces, right? And it loads pretty poorly on the controls and loads pretty poorly on the low visual stuff. Network number 2 also loads very highly on this navigation stuff and less so on the other non-navigation things. And network 9 is similar. These three all seem to be subnetworks that are biased toward navigation-related features rather than other kinds of features. So we can now project these three clusters onto the surface of the cortex again. It's cluster 1, cluster 2, cluster 9, and they're shown in red, yellow, and blue. Red, green, and blue, I guess. Red, yellow, and blue. I think that's just because the way Zhang labeled these colors to begin with. So you can kind of see the red network exists below the IPS, not actually in the IPS, below the IPS and above the temporoparietal junction. Also in the retrosplenial cortex and the medial wall. It's going to be the medial wall of the brain. And there are a few hot spots for this network up here in prefrontal cortex and down here near auditory cortex. Cluster 2 is this kind of reddish-yellowish stuff. It's kind of broadly distributed, but there's a giant blob representing this cluster right here near temporoparietal junction, a bunch of stuff in the supplementary motor areas, the prefrontal cortex, and up here anterior to RSC. And then there's cluster 9, which appears to be maybe more closely related to motor and somatosensory information, but is also represented here in the medial wall, and a bit down here in anterior to the visual system, and maybe down here in the motor, the bottom of the motor system. Okay, fine. So what are these? Well, you can now map them back onto the feature spaces and try to get a feeling for what they do. And this is, of course, we're not at the modeling stage, right? We already modeled. We modeled, we got our data. Now we're at the data interpretation stage. And in neuroscience, the data interpretation stage always involves a lot of squinting. So we have to squint at this and try to figure out what, we didn't build this brain, we just got whatever it evolved. So it's going to be kind of messy. This is what we have. We have what we're labeling a sensory navigation network, a sensory bias or visual bias navigation network that loads really high on low-level vision and a bit negatively on the motor system and negatively on some of the navigation variables. So you can see that this is basically a vision network. Right? Then the green, oops, whoa, what did I touch? That's weird. Okay. All right, so there's a motor network that you can see loads very highly on motor-related variables and very low on everything else. And then there's this abstract navigation network that seems to be distributed across all the navigation parts of the brain. Excuse me, not parts, navigation parts of the brain, across the navigation feature spaces. I'm sorry, I misspoke. All right, now, oftentimes when neuroscientists or MRI people show these kinds of maps, they imply that these are discrete regions of interest, but actually that turns out not to be the case. These three navigation networks are actually just biases in one large navigation network. And the way you can determine that is you take the three navigation networks and you do principal components on them. And if they were three independent networks, then we should find on this flat map that all of the colors are either red, green, or blue, and there's no mixtures of colors. But as you can see, in all of these regions in prefrontal cortex, below the IPS and on the medial wall, what we really have is gradients of colors. So these seem to be, there's this navigation system that has gradients of sensory motor information inside them. All right, let's see, what time is it? Oh, I'm doing okay. All right, so there are, once you're at the interpretation, excuse me, I shouldn't say that. There are other ways you can model these data rather than building an encoding model. So that's what my lab spends most of its time doing. And the young kids always want to throw a deep network at everything. If I was young, I would do that too. It's the most exciting thing going on. There are two kinds of deep network approaches you can take, supervised and unsupervised. I tend to be biased to unsupervised because supervised methods don't generally work well with neuroscience data because you don't have enough data to do the training. So there are a whole family of unsupervised methods that allow you to learn interesting structure in your data even if the data set is fairly small. And one of the most powerful of those is an autoencoder. One variant of an autoencoder is called a variational autoencoder. A variational autoencoder basically takes the training data and maps it into the brain data directly by essentially learning a low-dimensional subspace that links the training data to the brain data. So in this case, our training data only consisted of what the person saw visually while they were driving, and the brain data is just whatever the brain data was. So we build an autoencoder that gives us a low-dimensional feature space, and now we take that low-dimensional feature space and we can regress it times. Excuse me. So we learn the latent features. Now we have the latent features, and we use them in regression to discover a set of weights that weight each voxel on this low-dimensional autoencoder latent space. And now we can take the latent space, the tuning weights associated with an individual voxel or a region, and we can reconstruct what the stimulus was that we predict would have evoked the largest response from that voxel or that region. So this is essentially a way of doing decoding and trying to get a better intuition for what's going on in this data. Now, if you want to, you can also run model connectivity on this encoding, this variational autoencoder model, because we've kind of taken the VAE and shoehorned it into an encoding model framework. This is fine, but I don't think this is the most interesting thing that came out of this project. The most interesting thing was where if you actually look at individual networks, wow. That's odd. My computer has really been acting strangely lately. Okay, if you take some subnetwork from one of these networks they've recovered, like for example this light blue one, you can project it onto the cortex, and then you can decode what information was available, what information would have been predicted to have elicited a large response from that network or a small response from that network. So you can see that this higher order vision network here, the positive response seems to occur when there's another car in front of you, and negative response or low response occurs when it's an open road. So obviously, this car in front of you is more salient to visual information at the phobia than an open road. So that checks out. You can do this for a bunch of things. So if you notice, this hands representation is activated when people are, when they're turning, and there's a lot of horizontal optic flow, which happens when the car is turning. And this network is not activated when there's just low spatial frequency content, no optical flow that is rotating, just optical flow expansion, and the car is decelerating. So this is basically, if you're slowing down to a stop, this hand representation is just holding on the wheel. They're not turning the wheel, so you don't get any activity. You get a peripheral vision network, again, when you're high spatial frequency content and horizontal optic flow. So this is, again, this peripheral areas of the visual system are stimulated when you're turning. Why? Because when you're turning, you look where you're turning, and so you've now moved the representation of the world into the periphery of vision. Foveal vision, you know, this is all kind of what you would expect. Left foot, same thing. Now we can look at this navigation network. This navigation network, you notice it is activated during the go cue. The go cue is the cue that tells you where you are going to go. So obviously, the second you are told, drive to the gas station, you have to essentially retrieve from long-term memory the cognitive maps you will need to actually solve this problem from where you are to the gas station. So there's a lot of memory access and cognitive function that goes on during that time. Okay. All right, so the last thing I'm going to mention, this is almost the end, it has to do with attention. So in prior work from my lab and from a lot of other labs, it's been shown both in neurophysiology and in MRI that attention doesn't just change the gain or the baseline response level or the correlated noise in neurons, but it actually changes the way neurons represent information. And the reason for this is that neurons pool across levels of the deep network in your brain. So if you only have little tiny changes in the baseline response or the gain at one level of the brain. When you pool across neurons across multiple levels, that will actually shift the representation at subsequent levels. And the way this network is implemented in the brain, it implements kind of a matched filter mechanism. So if you're searching for vehicles, all of the neurons and all the voxels in your brain try to optimize themselves as much as they can to search for vehicles. They become vehicle detectors. And when you're searching for people, the voxels or the neurons become people detectors. Now, a neuron in V1 cannot become a person detector. It's just a Gabor function. So the amount that a neuron can shift its tuning in response to attention is directly proportional to the amount that attention influences the firing rate in an attention task. So in primary visual cortex, attention only has about a 2% effect on firing rate. In prefrontal cortex, attention has a 100% effect on firing rate. So if you look at these maps under different states of attention, you'll see in primary visual cortex, the maps really don't change at all. But in prefrontal cortex, if you're searching for people, all of prefrontal cortex, a bunch of it becomes a people detector. Searching for vehicles becomes a vehicle detector. Okay. This navigation task is an attentionally demanding task. So you would think that as you are allocating attention to different objects in the navigation task, that we should see these attentional shifts, and in fact you can. So the way we do this is we have people passively watch other navigation videos from our experiment when they're not navigating. And the video game engine gives us 16 different semantic categories of objects. So we can build a model that represents those 16 categories of objects, and we can map the representation of these objects across the surface of the cortex. We can do the same thing when they're navigating, because now they're just driving in the task. The video game is still offering us these 16 different semantic categories. So we can still build a navigation model, a semantic model, and map the representation of these semantic categories across the surface of the cortex. And you can just see right away that these maps look very, very different from one another, right? So during driving, the representation of these categories of objects in the brain is very, very different than if you're just passively watching the video. Now you can take the difference between them, and this difference kind of tells you what direction the representations are shifting in. And to make a long story short, it follows your intuition directly. When you're driving, the representation of driving-related features like roads, other cars, traffic signs, traffic lights is increased, and that comes at the cost of representation of all the other stuff you don't have to pay attention to. Now, we also know from prior work that if you have a passive viewing condition, vehicles tend to be represented in the place-related areas. So that would be the PPA, the OPA, and so on. And they're not, so you can see here, this is a map of vehicle weights. You can see PPA represents vehicles, OP, the occipital place area, the IPS and the RSC are representing vehicles. But if you now have people navigating, the vehicle representation completely changes. Now what ends up happening is the vehicle representation shifts. Now, instead of representing vehicles in the place-related areas, you represent them in the animate representing areas. So basically, you have this whole, your brain is well known as this whole network for social cognition that represents both the visual and social aspects of the social world, which makes sense because primates are social animals. So when you're driving, if you're not engaged in interacting with the other agents, the other vehicles, then a vehicle is just a mechanical object, and it's just represented in a whole constellation of semantically selective cortex that represents mechanical objects. The minute you have to interact with that vehicle as an agent, its representation now completely shifts, and it now is treated as another animate agent. So that basically the difference during active navigation between the representation of pedestrians and vehicles is very small. So you're completely shifting the way the brain represents information in this task from one whole complete subsystem, the inanimate system, to the animate system, quite dramatically. This is actually the biggest attentional effect I've ever seen in any experiment. And this is just another way to show the same thing. And so this actually shows you how this is inanimate versus animate. This is essentially we're projecting all, these are individual subjects, and these are the individual 16 semantic features, and we're looking at how these different features project onto this animate axis. And you can see that most of these features do not appear to be very animate, except for pedestrians, which are very animate, but when you're actively driving, these vehicles are also represented much more like an animate thing than any of the other semantic categories of objects in this task. Okay, so I told you that active navigation is supported by broadly distributed networks. This involves at least 3 sensory networks, 2 distinct motor networks, 2 or 3 networks that represent navigation-related information. The number of networks, remember, that you claim depends on where you cut the dendrogram, so it's kind of arbitrary, but this is a good first approximation of the number of networks involved in each of these functions in this task. I didn't discuss this earlier, but each of these feature spaces was designed to relate to a specific hypothesis. So here are all of our feature spaces. Here's a description of the feature space. Here's the number of features in each feature space. And all of these feature spaces are used in a series of hypotheses. So rather than thinking, oh, Jack did this weird, spooky coding model experiment, I don't understand it, what you can do is say, no, we did one experiment where we're testing all of these hypotheses simultaneously in one experiment. And to test each of these hypotheses, what we do is we instantiate a feature space relevant to that hypothesis and determine whether and how it's represented during navigation or differently during navigation versus passive viewing. Right? So we're engaged in this right now. There will be a separate paper that just lists all these hypotheses. These all mostly come from the rodent literature. There's some from the primate literature, maybe from the human literature, and we can test them all in one experiment. So the thing I really love about this framework is, I don't have to do 34 experiments to test 34 hypotheses. I can just do one experiment and test them all simultaneously at the same time. So I didn't really talk about this, but some of the known navigation-related regions of interest, like PPA and OPA, are actually divided into subregions that represent different kinds of information. You can also pull that out from this task. I did show you that active navigation leads to widespread shifts in semantic conceptual representations. The most important being that inanimate things, vehicles, become animate things if you interact with them as an animate thing. And then I showed you this VAE thing just because everybody always asks me to show decoding results. Okay, so where are we going with this? Well, the data that we have thus far suggests that we can probably get a better, more fine-grained idea of what's really happening in these navigation subnetworks, and we're working really hard on that. These are very correlated features. They're very abstract cognitive features. So it's a hard problem, but I think we can make progress on it. We have the data to do it. One really promising future direction is to not take features from just the stimulus and the motor response and the navigation goals in the rodent literature, but to actually take features from an autonomous vehicle. So we have 2 kinds of autonomous vehicles we built. One is an end-to-end learned autonomous vehicle that drives around in this environment, and the other is a principled autonomous agent that's built to do multi-agent interactions in a driving scenario, and it's just a piece of math. It's not a neural network, and it's a very explicit model, and we can fit both of those models to the driving data and pull out more subtle cognitive variables having to do with the way that people drive and the way they control the car during this task. And we also have a goal to model navigation in open arenas. So you may have noticed that there's a giant lake in this experiment, and turns out you can drive on that lake. So we can put obstacles in the lake, and then we can do an open arena mouse experiment where we have landmarks. And you just drive around in the open arena, and that's the next thing up to be done on this. And we can do that in the same subjects that did this task. Right? And finally, this general approach can be applied to any kind of open world task that you might want to do. All of the modeling software that's used here it's just our voxel-wise encoding model software. You can get it off my GitHub page. The tools to do model connectivity are on my GitHub page. The tools to do the voxel-wise encoding model fitting is a very nice toolbox that interacts very closely with scikit-learn called Himalaya. That's available on the GitHub page. And there's also a bunch of tutorials on how to do voxel-wise modeling if you want to bring this method into your own MRI workflow. Because you can do voxel-wise modeling in any MRI experiment. except probably a resting state functional connectivity experiment, because you don't have any X variables in that case. Right? So all of this is basically, I think, ready to be shared. It's ready for any person to be used. And if you decide you want to do this and you have questions, you can just email me at any time. That's it. Thanks very much for your time.
Great. Any questions for Jack? Question in the back.
Sorry, not exactly a question, but I have an animation about the brain that you wanted to show.
Oh, I will find that while I'm parallel processing the questions. Thank you very much. That might take us a second or two here.
I'll ask the question when.
Hugh, go ahead. I can parallel process. This is my list.
Great talk, Jack. I just wanted one speculation, really. Driving is a relatively recent evolutionary development. We obviously evolved, all multi-celled organisms pretty much, evolved navigation, presumably in a situation where you want to avoid static elements, run away from predators, and run towards food, or some notion like that. So how do you think this picture might change or might have evolved, given that driving is only 100 years old?
Let's see. Before we drove, we had horses. Before we had chariots. We had litters that people would carry other people on, right? We had piggyback riding. We had, you can ride a mastodon if you can lasso it. Right? It's not. I think most of the navigation stuff that's going on here is pretty generic, right?
But that's roughly 10,000 years of history, or 15,000.
Yeah, but other animals, I don't think the drive. I guess what I would say is don't get caught up in the driving aspect of it. We could have had people walking in this environment and not had anything to do with driving, and we could have done all the same thing. And all that would have happened in that case is we wouldn't have had controls, surfaces, and that would have been it. Everything else would have been exactly the same. But the Navy wouldn't have given us money for that. So, and you notice there's a little control surface in here. Why would somebody spend the months of work to build a stupid magnetic safe steering wheel and show that it doesn't cause MRI artifacts? Because Ford was paying us. And when we showed this off to Ford with a little control joystick, people don't drive that way. So, okay, fine, we'll build you a steering wheel. Want a steering wheel? We'll build you a steering wheel. Did it change the results? No. These things do not matter, right? So, you gotta kind of go up a level here. This is just navigation. It just happens to be instantiated in this driving environment. And I think the only thing that's special to the driving environment versus other things is this imputation of inanimate agency to inanimateness to animate agency that is, it's one of these results where it's what I call a type 3 result. Something you probably never really thought of before, but when you hear it, it's like, oh yeah, it kind of makes sense, right? Just like there's a long history in the primate literature showing that when primates use tools, they essentially become an extension of the primate's motor system, right? And this is kind of like that. We, the primates in our experiments, humans, are treating this vehicle as if it is a social agent, and therefore the relevant parts of the brain are the social parts of the brain. I think that's probably the one thing that you wouldn't get, just from like walking around, unless it was other pedestrians. Yes, hi.
Hey, much louder than I expected. Very cool talk, I really enjoyed it. I have one question just kind of about the model connectivity. Is it still looking at connectivity between brain regions over time in the same way that like your typical functional connectivity might be? Or is it just like kind of like pattern similarity between the model features? So you said you had all these different features and they had all different weights for a given voxel. So it's not necessary, those are derived from time, but it's like the pattern of contributions of all these different features is similar across two voxels. So it's kind of like they're operating the same way.
Yeah.
Okay, got it.
Believe it, You are the first person who's asked that question. I've been waiting for somebody to ask that question, so congratulations. It's a good question because it's not obvious the way I talked about it, or even the way people think about this, that functional connectivity, especially if you think about resting state, where there's no X variables, right? Something's driving resting state activity in the brain, but it's latent variables that you don't measure and you don't control. So you don't have any X variables. So the only thing you have is the Y variables. The Y variables are a time series, therefore functional connectivity lives in time. It's correlations over time. This encoding models, you can abstract out time if you want. You can expand time. But the first order encoding models, you've collapsed over time because you're building the model orthogonal to the time series. So yeah, that's a fundamental difference here. When we did the functional connectivity that I showed you, did the functional connectivity for a language experiment, we did the functional connectivity for model connectivity. Excuse me, we did the model connectivity. Let me try that one more time. We did the functional connectivity on the language experiment. We did the model connectivity in the language experiment. We did the functional connectivity in the normal way you do functional connectivity. So it only used the time series data. It didn't use the semantic features.
Sorry, I was just, I was kind of unclear for, maybe it was the task or the environment. Like, what was it exactly about the setup where you were able to distinguish between like an inanimate part that will activate like the PPA or whatever, and then the animate task? Because I would think like driving is like only the animate one. Are they seeing like still images of like other cars around them? It was like
well, okay, let's just show this. So here's the movie that I wanted to show at the beginning. If I can find where my mouse went, I will. It's got to be here somewhere. There's only so many places for it to go. Okay, there we go. So here's the movie I was going to show originally. We got a brain. It's inconveniently folded up inside the head, which is extremely annoying. So we extract it, put it on our computer, and inflate it like a brain-shaped football. And then we put some slices in it, and we flatten it out like a pizza. Okay. And so now we can actually look at the whole brain. And now at the top, you see somebody driving. It looks like the sound's off. There's actually sound. You can see somebody driving in this task. And at the bottom, you see the brain activity. Okay. So during this task, we know where the cars are. We know where your car is, and we know where all the other cars are, because the cars are autonomous agents in this Carla layer running on top of Unreal Engine. So we know in each frame, is there a car in the frame, is there not a car in the frame? And where is the car in the frame, and how big is it? And are you approaching it, or are you backing away from it, right? There are also, I don't know, it looks like this is Sunday morning or something, so there's not that many other things going on here, but there are also pedestrians in this environment. And the pedestrians will wander around on the sidewalks, and they will cross the street at inopportune times, so you have to avoid hitting them. And so we can actually map the representation of pedestrians in the brain, but just by regression. Every time there's a pedestrian, we can find out where in the brain was activated, right? So we have the pedestrian representation, the social representation of the brain. And what we see is, during passive viewing, the representation of vehicles and the representation of pedestrians is completely different. And it follows the classic distinction. The first principal component of MRI data in these kinds of vision experiments is animate versus inanimate. Right? And so the vehicles go in the inanimate networks, and people go in the animate networks. But when you're doing an active driving task, now the vehicle representation shifts, and they're represented in the same place as the people. Is that clearer? Hmm?
I think I saw a slide where there was a figure that mentioned non-goal directed navigation. Was that equivalent to the passive viewing task, or did you collect data during that familiarization phase when people were just exploring the environment, kind of aimlessly wandering? Because I'm curious what that would look like, whether people...
I actually can't remember what that feature space was. I'm sitting here like ruminating on it. I do not remember what that feature space was. But I have that data because I have the whole list of actually what the features were. So you can come find me afterwards. I can dig it out on my computer. Sorry, I don't remember which that one was. I'll just keep this, letting this play. Any other questions?
Yeah, just come on.
Yeah, great talk. Well,
Thanks.
what I'm curious about is when we're navigating, there's neurons that change our head direction cells. Like, we have these sensory signals that happen in our brain from the vestibular apparatus, which is not happening in this. So can you tell me what you speculate would be the story of this?
I speculate that reviewers will complain about that. And they already have. You know, the vestibular information is obviously important, especially when you're driving. You know, we've all had the experience of being car sick. Interestingly, you've also all had the experience that if you're driving, you are much less likely to get car sick than if you're a passenger. And this has to do with when you're the navigator, when you're the driver, you are looking where you're driving and where you're going, you're anticipating what's going to happen to the car, and so you have an anticipatory match between the visual input and what subsequently happens in the vestibular system. And if you're a passenger, that tends to be, that correlation is lower and it's broken. So clearly there was a big vestibular input to this system that we just are not looking at all here. And until somebody invents a portable MRI machine, something like that happen anytime soon, maybe when superconductors, you know, room temperature superconductors are invented, we could do that. But right now we can't build that machine, so we don't really have any way to look at
That
that.
would be cool. So one thing I was thinking about during the talk is you know where PIBC is in the map. And then so you could use that as a model predictor based on what the path was for that person. And you could see an inhibition or an increase in the bolt signal when people actually turn, because they know they're going to turn. They live their life in the real world.
Yeah, yeah.
And then you see a speculation of that, like in different copy,
Yeah.
what they were going to do when they're going to turn right.
Yes, yes,
That would
yes.
be super cool.
We are kind of looking at that because I'm glad you mentioned that. One of the neat things that you can do with MRI data is you can not just look prospectively, but you can look retrospectively, and you can find regions of the brain that are essentially anticipating the future. And the kind of most common use of that for people is in language processing, where people look at conversations, or they look at speech production versus comprehension, and you can see there's shifts in the networks having to do with anticipating what's being said versus understanding what's being said. Right? So you can also play that game here, and Zhang has just started to look at that, but so that will be coming out too. That will be another thing to come out of these data. This is like a data, you know, through a fire hose. So we're looking at that.
Hi, I really enjoyed your talk. So I was first of all wondering if the networks are modulated by performance. So errors, did you look at accuracy generally and maybe even strategy use? You mentioned that there's individual differences even in initial learning of the environment. So, you know, some people, I think you assumed that people are relying on some kind of cognitive map, but we know that some people actually take very different types of approaches to navigating. So I'm wondering, I guess it's two questions. One is generally if there's any relationship or prediction based on the network on whether somebody is making an error or not reaching a goal, and then also if there's any relationship with strategies.
Those are both great questions and hard, easy to answer in one way and hard in another. There are enormous individual differences in this task. There are bigger individual differences in this task than any experiment we've ever done in the lab. So people have very different strategies when they're driving. They have very different levels of, it's almost, there's different levels of coherence of the networks. Like some people have, you know, very clear networks that are pulled out of their brains, and they're easy to interpret. And our wonderful, brilliant Italian postdoc who refuses to follow the traffic rules has a chaotic navigation network that's, you know, where everything's just a mess. The data I showed you today, to compensate for that, was basically all the encoding model weights were aggregated at the group level in FS average space, and I should have told people that. And I don't have any individual data here, but it is quite diverse. So I think that diversity in these individual differences do reflect differences in cognitive strategies, exactly what you're talking about. The problem is right now we only have 6 participants, so that's not really enough to really understand what's going on with this. We would need a larger cohort. So this is something we're looking at. Also, my lab has a big project ongoing to better understand individual differences in MRI data at the single voxel level, and some of those new methods that are coming out will also apply to this data.
Well, what if the Italian postdoc relies more on his vestibular and inference compensing?
That may be true. Given his driving behavior, which is completely erratic. we all make jokes in the lab about, because for any of those who not driven in Italy, who have driven in Italy, you'll understand what we're talking about. We make jokes in the lab that it's because it's Italian, but it could be just that he requires vestibular input. I'll ask him about that when I go back in the lab.
Great talk, thank you. So in real life, after a while, we do most of the driving automatically without really thinking what we are doing. So at some point, there must be an automaticity involved, and those kind of automatic cognitive behavior usually depends on subcortical areas such as basal ganglia. So I'm wondering if you have any thoughts on not the cortical networks involved in navigation, but subcortical network involved in navigation.
I'm going to answer that. Before I answer that, I forgot to answer something over here that's relevant to what you just asked about. In this experiment so far, we've had people learn outside the MRI machine. They spend about 10 hours learning. But that's not actually what I think ONR paid us for. They want to look at learning to navigate. So now we have one participant who's actually spent 10 hours in the MRI machine learning to drive in this map. And we're going to try to start collecting more data for learning, so we'll be able to get at this issue. We'll also be able to get at your issue. But unfortunately, we're only going to be able to get at half of your issue. We can look at learning in the cortex, but the subcortex in MRI is really horrible structure to try to image. Subcortical nuclei are, for two reasons. First of all, subcortical nuclei are oftentimes located near air sacs and other ventricles and things that cause susceptibility artifacts, reducing signal to noise. But also, there's a square root of n distance law for measuring these signals in MRI, which is MRI is basically measuring electromagnetic emanations from the brain with antennas. And the farther the thing is that's emitting from the antenna, the lower your signal is. So MRI is great for the cortex, because that's all right next to the skull. It's great for vision, because it's back of the head and there's no air sacs there, right? MRI is really a thing built for vision experiments. Don't tell everybody else that. It really that's what it's So, but hippocampus, all the subcortical, the basal ganglia, the thalamus, those have horrible MRI signals. And so you just have to be very, very suspicious when you look at those signals in those regions. James.
Hi, Jack. Great talk. So at the end of the talk, you mentioned the possibility of using deep networks, and I guess the idea is to take those deep features to predict brain activity.
Yeah.
And
I don't find that particularly satisfying, but that's what the plan is.
Yeah. So I was thinking it would be nice, specifically for the visual areas. So in autonomous driving, deep network research, there's a lot of focus on object detection, but there are 2D object detectors and 3D object detectors, and so there's a suite of 2D algorithms like semantic segmentations to the extent of the image plane, but then there's monodepth algorithms which are trying to infer a range map from the image.
Right.
So I'm interested in whether you could do that, what you're suggesting, and identify to what degree these brain areas are really coding in the 3D world versus coding in the image plane, and I would suspect more.
Yeah. So we actually have some of those features in these data already, because we have the inferred 3D distance to the objects is one of the features that we mapped.
Okay.
So I mean, it's obviously it's a 2D projection. We didn't do it in stereo.
Yeah.
I never put any pressure on anybody in my lab to do stereo because I'm stereo blind, so it's irrelevant to me. But we know what the inferred 3D distance was, and distance to the objects is one of the very important variables in this task, both because it helps us understand the vision models, but also because it's important to understand the inferred 3D distance for navigation.
Mm-hmm.
So that's all in there.
So that's the
You could
distance
go dig it out.
to what in the...
I can't. I have to go dig it out.
Okay.
But
Okay.
if you email me, we can in 5 minutes I can tell you.
Yeah.
So my lab does a lot of brain viewers. If you go to the lab website, you'll see brain viewers. And now instead of just having kind of passive brain viewers, we're trying to shift to more active, participatory kind of viewers. So we're going to try to build a viewer for these data that will allow you to like drill down and look at how specific features are represented and specific feature spaces, and that's how you do it.
But from what you've done so far, can you say anything about the degree to which somehow the visual representation is in sort of 3D allocentric coordinates or egocentric coordinates versus like image plane processing? Or is that
Well,
something
given the way you posed the question, I will answer yes.
Okay, and could you elaborate?
Oh. I don't have that at the top of my head.
Great, so we're going to have a...
One more question in the back.
Oh, all right, one more question. And then we're going to have a reception where we can have lots of questions.
When you differentiate these animate and inanimate objects, do you think it can be just the representation of allocentric space in the brain?
Yeah, that's what James was just asking about.
Oh.
You look at allocentric versus egocentric. And weirdly, when we start, I should point out, when we started this project, that was kind of the main goal, because that is, you know, I was joshing with you, James, but we do take this seriously. One of the main goals is to look at the difference between allocentric and egocentric representations. We haven't done that yet because that's like buried in this hypothesis testing list of hypotheses, right? So as we go through that giant matrix of hypotheses I showed you, there are several hypotheses relating specifically to allocentric versus egocentric representations. And when we work through that list, that answer will pop out. So it won't be the first paper, but probably like the third or fourth paper out of this project will have the answer to that question. Okay. But I can't tell you the answer to that today. Zhang may have already answered that question. He just did not transmit that knowledge to me. And as all of those of you who run a lab or work in a lab know, the PI actually doesn't know anything. We're just lucky heads. He didn't tell me something, I don't know what happened. So that's the problem we have now.
Great, thanks so much, Jack. So I'd like to thank Jack again.